Today's mission on this deep dive is, well, it's to rethink pretty much everything you know about how your mind actually works.
Yeah, we are really turning the whole classical view of the brain completely upside down today.
We really are. We're using this groundbreaking new paper. It's from Nature Neuroscience. The title is Competitive Interactions Shape Mammalian Brain Network Dynamics and Computation.
Which is a mouthful, I know.
Right, totally a mouthful.
Yeah.
But the premise is just, it's fascinating because, we usually picture the brain as this perfect, harmonious symphony.
It's like a biological orchestra.
Exactly. Millions of neurons, dozens of regions, all, holding hands and firing together in this beautiful cooperative rhythm to generate your thoughts. It's a really comforting image. Total teamwork.
It is, but what if that assumption is literally missing half the picture?
Yeah. What if the real secret to the brain's genius, the thing that allows you to focus, to remember, to just be you, isn't harmony at all?
Right. What if it's actually an orchestrated battle?
That is exactly the shift in perspective we are exploring today. We're going to look at how scientists are literally rebuilding mammalian brains inside computers, right?
Using data from humans, macaques, and mice.
Yeah, to uncover this profound reality that our brains are wired not just to amplify signals, but to aggressively shut them down.
And understanding the competitive architecture, it really changes everything about how we view cognition.
Okay, so let's unpack this, because before we can even grasp this idea of brain competition, we need to understand how scientists are mapping and simulating the brain in the first place.
Right, because you can't just take a living brain out, put it on a desk, and, poke it to see how the network functions.
Yeah, that would be highly frowned upon.
Definitely. So to solve this logistical hurdle, researchers rely on two distinct ways of looking at the brain. The first is structural connectivity.
Which is basically the physical wiring, right?
Exactly. It's the anatomical wiring diagram. Imagine drawing a map of every physical cable connecting different hardware components in a computer.
Okay, that makes sense.
In humans, we map this using something called diffusion MRI. And what this technology does is it tracks the microscopic flow of water molecules in the brain.
Wait, water molecules? How does that show us the wires?
Well, water diffuses most easily along the protective sheaths of nerve fibers. So by tracking that water movement, we can mathematically reconstruct the major physical highways of the brain.
Oh, wow. So the water is like cars leaving a trail on the highway. You just map the trails.
That's a great way to think about it. And for the animal models in the study the mice and the macaques, they can get even more detailed.
Because they can use slightly more invasive methods, right?
They use chemical track tracing. They literally inject a specialized dye into one brain region and watch it travel physically down the axons.
So they can see exactly where that physical wire ends up.
Precisely.
Okay, so structural connectivity is the physical road network. It's the concrete highways, the dirt paths, the bridges connecting the different cities in your brain.
Which brings us to the second way we look at the brain, functional connectivity.
And this is usually captured with fMRI, right?
Yes, fMRI, which measures changes in blood flow. When neurons fire, they need oxygen, so blood rushes to that active area. We call this the BOLD signal.
BOLD, right. Blood oxygen level dependent.
Exactly. So with functional connectivity, we aren't looking at the physical wires anymore. We're looking at which regions activate together in time.
Like if region A in the front of your head and region B in the back light up at the exact same time, we say they're functionally connected.
Right, even if we don't know the exact physical wire that connects them.
Okay, so to stick with the visual, if structural connectivity is the physical road network, functional connectivity is the actual traffic pattern.
That's a perfect analogy.
You see this massive fleet of cars moving together from point A to point B, even if you can't see the specific sequence of physical streets they took.
And that actually sets up the big challenge for these researchers. It's something called generative computational modeling.
Right. They want to take that physical map of the roads, put it into a computer simulation, and see if they can recreate the real-world traffic patterns.
Exactly. And to simulate that traffic, they use a mathematical framework involving nonlinear Stewart-Landau oscillators poised near a critical Hopf bifurcation point.
Okay, hold on. Stop right there. I know we want to dive deep here, but we absolutely need an ELI 5 explain. Like, I'm 5 for that. Nonlinear Stewart-Landau oscillator poised near a bifurcation point. That sounds like a warp drive from Star Trek.
Yeah, fair enough. It is dense jargon. Instead of doing the complex math, think of these oscillators like physical pendulums.
Okay, pendulums, I'm with you.
Each brain region in the computer model gets its own pendulum. Now, A bifurcation point in dynamical systems is basically a fork in the road. It's a tipping point.
Like balancing on a knife's edge.
Precisely. On one side of this tipping point, the pendulum just hangs dead still, total silence. On the other side, it swings wildly and chaotically.
Oh, I see.
So by poising the mathematical model right on that knife's edge between silence and chaos, a tiny little push can make it swing in smooth, rhythmic waves.
Which is the perfect way to simulate neurons, right? Because they snap to attention, fire in these synchronized rhythms, and then quiet back down.
You nailed it. That's exactly why they use that specific model.
Got it. So you have these little mathematical pendulums, one for each brain region. You connect them inside the computer using the physical wiring diagram from the MRI. And then the computer tweaks the weights, like the influence of those connections, until the artificial traffic matches the real human fMRI traffic.
That is the core mechanism. The algorithm just iteratively updates the weights of those anatomical connections, hunting for the perfect formula.
But historically, these old models had a massive blind spot, didn't they?
A huge one. They operated on the assumption that all long-range connections were purely cooperative.
Right, because the physical mapping tools, like the water diffusion MRI, they only measure the presence of a pathway.
Exactly. You can't look at a physical cable in an MRI and inherently know if it's sending a go signal or a stop signal.
So the older models just assumed everything was a green light. If region A fired, it automatically told region B to fire.
But dynamical systems in nature simply do not work that way. I mean, if a system only has positive, amplifying feedback loops, what happens?
It just spirals out of control, right? Like a microphone squealing near a speaker.
Exactly. In the brain, pure cooperation without regulation looks like a massive seizure. The network needs feedback control.
So the researchers behind this Nature paper decided to introduce a new parameter.
Yes. They allowed the algorithm to use competitive or negatively signed interactions.
Meaning the model could decide purely based on what matched the real brain data best, that when region A fires, it actually suppresses region B. It's a net negative influence.
And the results were just profound. Across all 3 mammalian species, the models organically chose to make a massive portion of the connections competitive.
Like how massive?
For humans, it was about 25%. For macaques, it went up to 38%. Wow.
And the model found these negative valued connections in the inferred connectivity of literally every single individual brain they tested.
Okay, wait. I have to put my skeptic's hat on here for a second. We've known about inhibitory neurons forever, right?
Oh, absolutely.
There are millions of GABAergic neurons whose whole biological job is just to stop other neurons from firing. So how is a computer model discovering competition a new scientific breakthrough?
That's a really crucial distinction to make. We have to separate the microscopic cellular level from the macroscopic whole brain level.
Okay, explain that.
You're completely right that local inhibitory neurons exist. But this model is capturing a macroscopic net negative effect between massive brain regions that are centimeters apart.
Oh, I see.
It's completely agnostic to how that suppression is biologically implemented on the ground level.
So you're saying it's not just one long inhibitory wire stretching all the way from the front of the head to the back? No, the biology is far more complex than that. The suppression might actually be caused by a long-range excitatory projection, a go wire, that travels across the brain.
But then what does it do when it gets there?
Well, it might happen to plug directly into a local cluster of inhibitory neurons at its destination. So the moment the go signal arrives, it triggers the local brakes, shutting the whole region down.
Oh, wow. So a go signal activates a stop mechanism.
Exactly. Or, it could be related to complex conduction delays where signals arrive out of phase and just cancel each other out.
Like noise-canceling headphones.
Precisely. Or it could even be related to how cerebral blood flow is dynamically shunted away from one region to supply another.
Okay, so the computer model doesn't care about the biological how. It just proves mathematically that the real-world outcome of that specific highway is active suppression.
You got it.
All right, so let's do a quick recap here because we've covered some dense ground.
Good idea.
Number 1, scientists use computer models to predict brain traffic based on physical roadmaps.
Right.
Number 2, old models only allowed for green lights, pure cooperation. And number 3, this new model proves that to match a real living brain, we need up to 40% of the network to act as a red light, a stop signal. The necessity of this orchestrated battle is just written directly into the math.
Which naturally brings us to the next big question, right? If a third of the connections in a mammalian brain are actively playing defense, where exactly are they located?
Yeah, are they just sprinkled randomly?
Not at all. The researchers analyzed the spatial arrangement and found a highly consistent architecture across humans, macaques, and mice.
Okay, so what does that architecture look like?
While the cooperative green light connections tend to be strong in magnitude, they are highly modular, and they are overwhelmingly local.
Meaning they connect regions that are physically right next to each other.
Exactly.
But here's where it gets really interesting, because the competitive connections are the exact opposite.
They really are. They are weaker, they're more diffuse, and crucially, they are long range.
They connect regions that are on completely different sides of the brain.
Yeah, and the wildest part is what they're connecting. Across all three species, these long-range competitive ties preferentially link brain regions that have completely opposite biological profiles.
Okay, opposite profiles is a massive concept here. What does that actually mean?
It means the model is building competitive links between regions that have entirely different cellular architecture, totally different gene expression patterns, and completely different densities of neurotransmitter receptors.
So think of the brain like a massive corporate office building, right?
Okay, I like this.
The cooperative local connections are like the immediate team chatting on a localized Slack channel. They sit in the same department, they have the same background, they do the same job.
So they generally agree with each other and amplify each other's ideas.
Exactly, to keep their specific project moving. But the competitive long-range connections, those are like the CFO on the top floor auditing the creative marketing department in the basement.
That's a perfect analogy.
Right, because they have completely different profiles, different backgrounds, entirely different priorities. Their job isn't to agree. Their job is to apply friction, to audit, and to actively suppress certain activities so the overall company doesn't go off the rails.
Yeah, I mean, that perfectly illustrates a recognized principle in network theory called the strength of weak ties.
Oh, right.
In complex systems, these weak, diffuse, long-range links are absolutely essential for global integration. They prevent the local, highly cooperative departments from just operating in total, isolated echo chambers.
They orchestrate the global state by applying that necessary friction.
Exactly.
So we have this beautifully complex spatial map of local cooperation and long-distance rivalry. But, the ultimate test of any computer simulation is how well it actually mimics reality.
Right, the fit to the data.
Yeah. When they added this 25 to 40% competition, did the artificial brain act more like a real human?
Dramatically so. I mean, the model's ability to recreate empirical, real-world brain activity just skyrocketed. The group fit correlations jumped up to 0.95.
And for context, 1.0 is a perfect match, right?
Yes. So 0.95 is an incredibly tight alignment between the simulated traffic and the actual fMRI brain scans.
Okay, but wait. I have to ask a foundational data science question here.
Go for it.
If you just give a computer algorithm more variables, like, say, allowing it to use negative numbers on top of positive ones, doesn't it automatically get better at matching a graph just by sheer mathematical flexibility?
That's a very valid concern.
Like, how do we know it's not just overfitting, meaning it's just memorizing the random noise of one specific person's brain scan rather than learning actual biological rules?
That is the exact trap researchers have to avoid. But here's the thing. If the model were just overfitting to random noise, it would perfectly memorize the original subject's brain scan, but it would completely fail if you applied its output to a different person's brain.
Because my brain's random noise is different from your brain's random noise.
Exactly. But the data showed the exact opposite. When they added competitive interactions, the model actually improved its generalization to other brains. Its self-other similarity went up, so it learned true, generalizable biological rules.
But this is the absolute kicker for me. It improved its match to the original brain even more.
Yes, it did. The self-similarity outpaced the self-other similarity. In the literature, this is known as differential identifiability.
Which basically means the addition of competitive interactions made the model significantly more subject-specific.
Yes. Your brain model became uniquely identifiable as you.
And we really need to explain why that happens. Like, why do negative connections capture individual identity better than positive ones?
It's a great question.
Because by actively suppressing certain connections, the brain forces information to take specific routed pathways rather than just flooding the whole system.
Right.
Think about water flowing down a mountain. If there are no dams or rocks, the water just washes over everything uniformly. But if you place strategic dams, which are the competitive suppressions, The water is forced to carve a highly specific riverbed.
And that unique routing, which pathways your brain actively chooses to block, is what forms your highly specific neural fingerprint. That is a brilliant way to visualize it. So the spatial architecture is mapped and the individual fingerprinting is verified.
Right.
But a living brain isn't just a static map, right? It is a dynamic engine operating across time. The flow of thought requires constant shifting movement.
So how do they evaluate that?
To evaluate how competition affects this temporal flow, the researchers analyze something called metastability.
Right, and they measure this using the Kuramoto order parameter, which, again, is another intense term we need to break down.
It really is. The Kuramoto order parameter is essentially a mathematical measure of how synchronized a system is at any given moment. In the brain, it tracks how regions alternate between periods of intense synchronization, where lots of areas fire in unison, and desynchronization, where they operate independently.
So imagine being in a giant sports stadium. Synchronization is when the entire crowd is doing the wave together. Everyone is doing the exact same thing. Desynchronization is when everyone sits down and starts having independent, localized conversations with the person next to them.
Exactly. And a healthy brain needs to do both of those things fluidly. It needs to globally integrate information, but also locally segregate tasks so things don't just get blurred together.
So what happened when they ran the old model? The one forced to only use green light cooperative connections.
The metastability was completely unrealistic. The artificial brain synchronized way too much. It essentially locked up in a rigid state of positive feedback.
So the whole stadium was just doing the waves forever, and no one could ever sit down to have a private conversation.
Exactly. It was useless for complex tasks. But introducing the competitive interactions moderated this behavior. The competition acted as a crucial dampener.
It kept the brain in a healthy, dynamic balance.
Yes, allowing it to effortlessly slide between global integration and local segregation. And furthermore, this competition fundamentally altered the architecture of information flow by creating hierarchy.
And this makes total sense because suppression forces asymmetry, right?
How so?
Well, if two regions are just mutually cooperating, they are just shouting back and forth equally. But if region A actively suppresses region B, region A becomes the clear sender and region B becomes the receiver.
Right, that creates a definitive top-down hierarchy.
It also increases what the researchers call synergy.
Which means information isn't just being redundantly copied everywhere. Different regions are forced to combine distinct pieces of information in unique, synergistic ways.
To prove that this mathematical synergy actually relates to real human cognition, they utilized neurosynth, right?
Yes, Neurosynth. It's a massive database containing 14,000 fMRI studies, covers 123 different real-world cognitive operations.
Things like memory recall, visual attention, fear processing, language comprehension.
Exactly. So they tested whether their computer model, just running its spontaneous resting state simulations, naturally recreated the specific traffic patterns associated with those actual cognitive tasks.
And what did they find?
The models with competitive interactions were significantly more capable of co-activating the exact regions that belong to the same real-world functional circuits.
And the cooperative-only models failed to do this effectively.
They did. If we connect this to the bigger picture, your brain absolutely needs competition to stop the runaway noise.
Right.
You need a massive portion of your brain to actively stay quiet so that the specialized regions, the ones trying to, say, recall a childhood memory or calculate a math problem, can actually be heard over the background chatter.
So without the competitive ties, the specialized functional circuits just get drowned out by global cooperation. Competition enables cognitive clarity.
Exactly.
Okay, let's do another quick synthesis for everyone listening because we are deep into the weeds here.
Sounds good.
1. We've uncovered that up to 40% of the brain's network acts as a stop signal. 2. These competitive links run entirely different opposite brain regions across long distances to apply systemic friction. 3. Your specific pattern of deploying these long-range sanctions creates your unique neural fingerprint. And 4. This active suppression prevents your brain from shouting all at once, allowing for complex, hierarchical thought.
Which perfectly sets the stage for the ultimate and honestly, arguably most futuristic test in this study.
Oh, I love this part.
Right. If this newly discovered blueprint truly represents the optimal way a mammalian brain computes, could we use this exact wiring diagram to build a better artificial intelligence?
Yes. The field of connectome-based neuromorphic computing. This part of the paper bridges neuroscience and computer engineering. They literally built an artificial neural network specifically, a reservoir computer.
Right. And for those unfamiliar, a reservoir computer is a very specific type of neural network. It consists of an input layer, a complex interconnected middle layer called the reservoir, and an output readout layer.
And the unique thing about reservoir computing is that the connections inside the reservoir are fixed, right? They don't change or learn during training.
Exactly. You just feed signals into it, and the internal network reverberates, creating these complex patterns. The AI only trains the output layer to interpret those reverberations.
Think of the reservoir like a pond. You toss a rock in, that's the input. The rock creates a wildly complex pattern of ripples interacting with each other. The AI's job is simply to look at the edge of the pond and figure out where and when the rock was thrown just by reading the ripples.
That's a great visualization. And what the researchers did here is they built the pond using the exact generative connectivity diagram discovered from the human and animal brains.
They literally used the biological blueprint, complete with its specific balance of local cooperation and long-range competition.
Yes. They used it as the AI's internal architecture, and then they gave this biologically inspired machine a specific cognitive test, a memory task.
Okay, how did that work?
They inputted random signals into the artificial nodes that corresponded to the brain's visual regions. Then they asked the artificial nodes corresponding to the somatomotor or movement regions to reproduce a delayed version of that exact signal.
So they were basically testing if the artificial brain could catch a visual memory, hold it in its reverberating ripples over time, and accurately pass it to its motor regions to execute an action.
Exactly. And the results were just mind-blowing. Across all three species, the artificial reservoir built with both cooperative and competitive interactions significantly outperformed the network built with purely cooperative ties.
I wasn't even close, right? It exhibited far greater memory retention and accuracy.
Much greater. The network physically possessed higher computational capacity.
That is just wild. We aren't just looking at colorful fMRI scans anymore to say, oh, that looks biologically accurate.
No, we are literally extracting the wiring rules of human and animal brains, specifically their long-range competitive sanctions, and using them to build better, faster, higher-capacity problem-solving machines. It's a profound validation of the model. It proves empirically that these long-range competitive ties aren't just a mathematical quirk designed to fit fMRI data.
Right.
They are a fundamental structural requirement for high-level memory and complex computation in literally any network, biological or artificial.
So what does this all mean for you listening right now?
Yeah.
Well, your mammalian brain is not a utopian harmony. It's a carefully orchestrated battle.
The balance between local cooperation and global aggressive competition is the absolute engine of cognition.
Yeah. Next time you are sitting in an incredibly crowded, noisy coffee shop, and you somehow manage to tune out the espresso machines and the overlapping conversations just to read and comprehend a single sentence on your laptop. Remember, that's not just your focus turning on.
No, it's not.
That is your brain physically deploying a massive network of long-range diplomatic sanctions to shut the rest of your mind down. Your genius relies entirely on your brain's ability to violently ignore things.
Which, raises an incredibly important question, something really vital to ponder as we step away from the data and look toward the future of medicine.
What's that?
If healthy, everyday cognition relies so heavily on this delicate, long-range competition, could the disruption of these weak competitive ties be the hidden root of severe neurological disorders where the brain loses its functional balance?
Wow. That shifts the entire paradigm.
It really does.
Because what if the key to fixing the brain in certain cognitive or psychiatric diseases isn't about finding the regions that are broken and failing to fire?
Exactly.
What if the problem is finding the distant regions that simply forgot how to compete? Maybe we don't need to restore the harmony at all. Maybe the future of neurology is learning how to restore the orchestrated battle.